This is Protein Synthesis by Uldis Bagley. This is the key which will be used in place of labels in many instances throughout the video. The beginning process is the same as DNA replication. Just blur that out, the DNA replication part, but it's the same. This is a blow up of this image here, this gene, and this gene is a flattened version of the twisted helical structure. Okay, so what happens is the helicase enzyme goes up here and breaks apart the hydrogen bonds between the bases, and then we get two separated DNA strands, as we see here. Now this is a blown up version of one strand, a half strand, and from this we get a 3 to 5 sense strand, where the DNA polymerase moves from 3 to 5. The DNA polymerase attracts free mRNA nucleotides, and they propagate along here to create a messenger RNA. Okay, so once the two are formed, the helicase comes back and splits them apart, breaking all the hydrogen bonds, and then we have the separated messenger RNA, which leaves the nucleus through the membrane, and that's the end of transcription phase during protein synthesis. Okay, when it leaves, it goes to a ribosome, and this is the catalyst for the next reaction. Okay, so when it's on the ribosome, amino acids come along. We've got examples here like asparagine and glutonic acid, and they connect to the groups of three bases, which are known as codons, and they join to the mRNA, and this causes propagation of the amino acids. And the top of the amino acids, we have the peptides, and they bond, and as this propagates, we get a propagation of peptide bonds, and we get a polypeptide group. And once this is terminated, when it gets terminated by a specific codon group, the tRNA, which is this part here, the codon and the bit in between, move away. The polypeptide moves away, and we get this, which is a polypeptide chain, which moves around the cell and can be changed into different proteins. It forms around, changed into different proteins. It can join with other polypeptides and form other proteins for different functions as well.